In this video, we will discuss about this CRT silenced pancreatic cells shows partial reduction in EMT pathways. In the previous video, we have seen that they have shown the bright field images of this pancreatic cancer cells, that is AsPC-1 and this Capan-2 that are treated that are induced with EGF. So in the scramble control cells, they can see the change in the phenotype, whereas in the CRT silent cells, the change in the phenotype is very minimal or reduced. So, and in the previous video, I have also said that they could have shown more detailed, more further analysis through this immunofluorescence with this EMT marker or through the cell signaling pathways. So in this video, we will discuss the next figure where they have checked the different signaling pathways that are affected by EMT. So my criticism is not exactly correct, as they have shown the results in the next figures. So here they have targeted the three different signaling pathways, that is integrin signaling pathways, EGFR-ERK signaling pathways, and this MAPK signaling pathways. And what they are able to see, that in the case of scramble, there is an increase in these signaling pathways, that we can see that integrins is increased, and also this other signaling pathway, this MMP9, this MMP9 and these vimentins, all these are increased in the case of this scramble. Scramble is the last column, where we can see that fibronectin, integrin, ERK signaling pathways, and Cathrin, vimentin, all these are increased, which is the markers for the mesochemical state when the scramble cells are treated with EGF. However, for the SG-CRT1 cell lines, they are able to see partial increase in these signaling pathways, and for example, this integrin beta 1 is increased partially compared to the scramble case, and also ERK1, except for the N-Cathrin, as N-Cathrin's increase is N-Cathrin decrease is not statistically significant compared to scramble. So these left-hand side results are for the Capan-2, and the quantifications are also shown here, where we can see that there is a increase in this fibronectin and integrins, C-mycin, ERK signaling pathways. However, for the N-Cathrin, they do not see a significant increase or a significant decrease in the CRT silence conditions compared to the scramble control conditions. And they are able to see similar results for the AsPC-1 cell lines also, which is also shown here, and it follows the same format, that is last column represents the scramble cells, which shows the increase in the fibronectin, integrins, basically the signaling pathways, that basically the genes that are involved in the signaling pathways of integrin, EGFR and ERK, and also in the MAPK signaling pathways. And their quantifications are also shown here, where we can see that there is an increase which is statistically significant for the scramble case compared to the CRT silenced cells, except for the N-Cathrin. And they have also did the similar experiment on the DXPC3 cells, but those results are not shown in the main figure, but their results, however, their results are in the supplemental figures. If someone wants to see those results, they can go through the supplemental figures. So from these results, and also from the previous bright field images, they made a point that the CRT silenced pancreatic cancer cells are affected, or the CRT plays a role towards this EMT transition of these pancreatic cancer cells in the EGF induced condition. So, and in the absence of CRT, those EMT transitions will also get affected. So this is what they have concluded so far. If you need to understand more about these experiments in detail, and go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.